If there is more sodium bicarbonate in the presence of the spinach leaves, then the faster the rate of photosynthesis will be. When there is a higher concentration of sodium bicarbonate, there will be more carbon dioxide present. This is because carbon dioxide is one of the components in sodium bicarbonate. Carbon dioxide diffuses through the stomata, and then eventually moves through the mesophyll cells into the chloroplasts. In the chloroplasts, photosynthesis occurs as carbon dioxide combines with water to form glucose and oxygen. Additionally, as the concentration of sodium bicarbonate increases, the concentration of carbon dioxide also increases. This means more carbon dioxide is available for many cells to undergo photosynthesis, as long as there is sufficient amount of water. More cells can produce glucose and oxygen under a short amount of time. Subsequently, more leaves will float, as additional oxygen increases the buoyancy of the leaves. In essence, when more sodium bicarbonate is present, the faster the rate of photosynthesis. Spinach leaves, 8 grams of sodium bicarbonate, 1 gram being used in trial 1, 2 grams being used in trial 2, 5 grams being used in trial 3, and trial 4, there will be no sodium bicarbonate as trial 4 is a control experiment. In a 200 ml beaker, 5 ml of liquid dish soap will be dissolved in 50 ml of distilled water. In each of the 4 200 ml beakers, 100 ml of distilled water will be measured. 3 weighboats for the sodium bicarbonate in trials 1, 2, and 3. A timer, a hole puncher, 4 10 cc syringes, LED ceiling lights, 4 stirring rods for each trial, scupula to measure the sodium bicarbonate, a dropper, a graduate cylinder to measure volumes, a weighing machine to measure the grams for the bicarbonate, a ruler, pen and marker for labeling, and masking tape. Prepare the lab station in a room where there are ceiling lights. This will be the light source in your experiment. Obtain all materials and apparatus, and wear necessary safety equipment, including an apron and safety goggles. Label the beakers appropriately. 4 200 ml beakers should be labeled as 1 gram, 2 grams, 5 grams, and 0 grams of sodium bicarbonate respectively. The 5th 200 ml beaker should be labeled as diluted soap solution. Using the hole puncher, punch out 30 discs of the spinach leaves while being careful to avoid the veins. Set the discs aside. In a graduate cylinder, measure exactly 50 ml of distilled water. Then, in the diluted soap solution beaker, make a diluted solution of the liquid dish soap. Using a pipette, add 5 drops of the dish soap to the measured 50 ml of distilled water. Using a stirring rod, gently stir the soap with the water. Be careful to avoid stirring too fast and making suds. Using a scupula, drop 1 gram of sodium bicarbonate inside a weighboat. Place the weighboat on top of a weighing machine, adding and removing sodium bicarbonate as needed, so exactly 1 gram is present. Using graduate cylinder, measure exactly 100 ml of distilled water. Add the 100 ml of distilled water in the 200 ml beaker labeled 1 gram of sodium bicarbonate. Then, add the 1 gram of sodium bicarbonate inside the beaker. Stir the bicarbonate in the water until fully dissolved. Using a pipette, add 1 drop of the diluted soap solution to the bicarbonate solution. Stir the contents gently, avoiding the formation of suds. Pull the stopper out of the bottom of the syringe. Place 10 leaf discs inside the syringe and pull in 5 ml of the bicarbonate in soap solution. Aim the tip of your syringe upwards and carefully push the air out, avoiding the crushing of leaves. Tap the syringe so air bubbles are released out of the syringe. Create a vacuum by completely covering the tip of the syringe with your finger. Draw back the plunger for 30 seconds. Release the vacuum so the leaves can be submerged into the solution. Repeat this 2 to 3 times until all leaves are submerged in the solution. Once the leaves have sunk in the solution, remove the stopper and quickly empty the syringe back into the sodium bicarbonate solution. This is to avoid the leaves having contact with the air. Using a timer, observe how many discs float on the surface of the solutions and the time intervals given in data table 1.0. Repeat steps 5 to 11 for 0 grams, 2 grams, and 5 grams of sodium bicarbonate with their respective beakers. Record all observations in data table 1.0. The cuticle on top of the leaf is hydrophobic, which means it repels from water, and it prevents the leaf discs from ever sinking. The soap is amphiphilic or hydrophilic, because it is essentially a fatty acid. It will coat the leaf discs. Then, this will allow the disc to sink after the gas is sucked out. This is important because we want the leaves to be fully submerged in solution and not exposed to any air. When we place the leaf discs in the sodium bicarbonate solution, the leaves will sink. Then, we will observe when the leaves will rise due to the production of oxygen due to photosynthesis. The reason we create a vacuum is like the reason we use soap. This is to get rid of oxygen, allowing the leaves to be less buoyant before we add them to the sodium bicarbonate and soap solution. Once we add the leaves to the solution, they sink. As photosynthesis occurs, the oxygen produced will cause the leaves to rise. We receive better results because the rising of leaves is due to the production of oxygen. We use sodium bicarbonate because sodium bicarbonate dissociates to carbon dioxide. The carbon dioxide is a reactant in photosynthesis, which reacts with the other reactant, water. This causes glucose and oxygen to be formed. Oxygen will cause the leaves to be buoyant and rise to the surface of the solution. If we compare the amount of leaf discs floating in a 20-minute time interval, we can determine the effect sodium bicarbonate has on the rate of photosynthesis. In trial 1, there was no sodium bicarbonate added. This resulted in no leaves floating. In trial 2, only 1 gram of sodium bicarbonate was added. This resulted in only 1 leaf floating. In trial 3, 2 grams were added, which resulted in 2 discs floating. And finally, trial 4, 5 grams of sodium bicarbonate were added, which resulted in 10 discs floating. According to the results, the hypothesis was proven correct because as the amount of sodium bicarbonate increased, the faster the rate of photosynthesis became. This is because the amount of leaf discs floating is dependent on the amount of sodium bicarbonate being added. Sodium bicarbonate dissociates to carbon dioxide in an aqueous solution, and carbon dioxide reacts with water to produce oxygen and glucose. Oxygen is one of the products of photosynthesis, so if there is more product, the rate is faster. In this experiment, it was determined that all 10 leaf discs floated when 5 grams of sodium bicarbonate was added. However, when there was only 1 gram of sodium bicarbonate added, only 1 leaf disc floated. As more sodium bicarbonate is added, the rate of photosynthesis becomes greater. In the case of no sodium bicarbonate being added, there was no leaf discs floating because photosynthesis did not occur. This is because carbon dioxide is not present, therefore a reactant is not present, so the reaction cannot occur. This results in no formation of air bubbles, and the formation of air bubbles allows the leaf discs to float up to the surface, which is proof that oxygen is not present. Experimental error is the leaves were being stuck at the grooves in the sides of the beaker, which prevented the leaves from floating up to the surface. This led to a discrepancy in results because not all the leaves were rising, and it indicated that the rate of photosynthesis was slower than it really was. A way to improve the experiment is to tap the areas where the leaves may be stuck, which will encourage the leaves to rise in the presence of oxygen. Another experimental error is the inconsistency in light. The intensity of light must be controlled, as using the ceiling lights are too far from the beakers and not directly giving the leaves light. Some beakers may have higher light intensity than other beakers. The light intensity is usually defined as the energy hitting an area over some period of time. So as light intensity increases, the rate of photosynthesis will also increase as long as other factors, such as enzymes, are in adequate supply. So in this case, more photoexcitation will occur in the leaves, resulting in more light-dependent reactions. This will yield more products, so it will give you more oxygen, which will increase the buoyancy for the leaves, and more leaves will be at the surface of the solutions. So for more accurate results, the light intensity should be controlled so the rate is not faster or slower than it really should be. Another way to improve this experiment is to run the experiment for a longer period of time. There was an experimental error by running trials for only 20 minutes, as some leaf discs could have risen to the surface if more time were given. For example, for trials 2 and 3, more leaf discs could have risen as some of the leaves were semi-floating at the 20-minute mark. If the experiment was observed longer, then there may have been more leaf discs floating to the surface, which would have altered the rate. In order to improve the experiment should have been run longer. The first challenge of a self-designed lab is designing a procedure. It was difficult designing a procedure as there are many things we must account for in a procedure. A procedure should be detailed and clear enough for someone who is unfamiliar with the topic can proceed with the experiment without much difficulty. However, the person designing the procedure is already familiar with the experiment, so they may make assumptions about what the reader may know, which may not always be the case. For example, it is important to indicate which measurement tools should be used to weigh the substances. Details like these are critical to implement, as a mistake in measurement can lead to an inaccuracy in results. For this reason, the procedure should be step by step with all the details present for accurate and precise results. The second challenge is controlling the variables in the experiment. It is difficult to determine which variables should be controlled for the experiment to be completed with accuracy. If there are too many variables being altered, there will be an impact on the dependent variable. For example, in this experiment, it is important that only one variable is impacting the dependent variable. The dependent variable in this case is the amount of leaves floating at the surface, and the independent variable is the amount of sodium bicarbonate being added. However, if, for example, the light intensity were not controlled, then the experiment will be immensely affected. Now, the rate of photosynthesis will be affected by light intensity and concentration of carbon dioxide, not just carbon dioxide alone.